This video discusses the mechanism of CRISPR-Cas9 in its natural context, bacterial immunity. Bacteria face routine harassment from bacteriophages, and sometimes they eat foreign DNA elements like plasmids. This can be harmful, and therefore bacteria often needs some form of immunity against them. Immunity, as you may already know, can be either innate or acquired through adaptation. Typically, a bacteria makes restriction enzymes, toxins, and has many other pathways to defend against pathogenic DNA elements. All such pathways are an innate response because they are unbiased and do not require any memory. Adaptive immunity, on the other hand, requires memory. Us humans have a range of complex immune cells that help us adapt. Vaccines are a way to create immunogenic memory. But we are complex multicellular organisms. How then can single-cell organisms like bacteria create immunogenic memory? The answer is in the DNA sequence of the invader. The DNA sequence of the invader can be stored as a memory in the bacterial genome at a specific location and retrieved at a later stage to defend against that same pathogen. The CRISPR-Cas9 locus is that specific location where foreign DNA sequences can be stored and retrieved when needed. Here is the structure of CRISPR-Cas9 immunity locus. It has 3 operons. One contains instructions to make a single non-coding RNA called tracrRNA. The second is a Cas operon, which codes for 4 proteins named Cas9, Cas1, Cas2, and CSN2. The third operon is called the CRISPR locus, which starts with a leader sequence that also has the promoter, followed by repeats. And in between the repeats are unique spacer sequences. All repeats follow the same direction, meaning that they are direct repeats. And each repeat is identical in sequence. In contrast, each spacer is a unique sequence. The length of both repeat and spacer ranges from 20 to 50 bases. The repeats often have a low level of palindromicity. Therefore, the CRISPR locus is essentially a library of repeats and spacers. And the unique spacers are the DNA sequences derived from pathogens, and they are physical manifestation of the bacterial memory. Hopefully, this short summary also shows you why CRISPR is called a cluster of regularly interspaced palindromic repeats. With that, let's make our way into bacterial immunity to understand how CRISPR-Cas9 actually works. There are 3 steps to this process. When a foreign DNA enters the bacteria, the first step in adaptive immunity creates the memory of the invader. In this step, a spacer is acquired, meaning that a small piece of DNA from the pathogen is taken and inserted into the CRISPR locus. After creating this memory in the form of a DNA sequence, the bacteria retrieves this memory from the CRISPR locus, where the CRISPR locus is converted into an RNA, and with the help of tracrRNA and Cas9 protein, an active nuclease complex forms. In the final step of interference, this active nuclease is used as a weapon to destroy the target DNA based on the matching spacer DNA. And this causes the foreign DNA to be digested. The acquisition of specific DNA sequences leads to genetic adaptation or immunity against any future infections by that same pathogen. There is something I need to clarify before moving further. There are 2 broad classes of CRISPR systems, and each class is subdivided. Among many differences, one big distinction is that Class 1 uses multi-effector proteins, whereas Class 2 uses a single effector protein. Effectors are just nucleases, and Cas9 is one such example. Cas9, by the way, is found in Class 2, Type II system. The Type II system has further subclasses that I won't discuss here, but it is one of the most widely studied systems and also used in genome editing. Therefore, this video about CRISPR-Cas9 is specifically on Type II-A. With that, we are ready to explore the 3 steps of CRISPR-Cas9-driven immunity and its mechanism. We start with memory creation, and it starts as soon as the foreign DNA tries to replicate itself. During DNA replication, the replication fork may sometimes pause. In those cases, a bacterial protein complex called RecBCD tries to fix this paused replication fork. Foreign DNA looks quite different from the bacterial DNA. So, RecBCD tends to overdo its job when fixing the paused replication fork, meaning that the helicase and nuclease activity goes a little out of control. Through some unknown mechanism, possibly involving other nucleases, parts of foreign DNA are indiscriminately chopped up. These chopped bits become the source of the next step, where a protospacer is created. In this, the Cas9 protein and tracrRNA from the tracrRNA operon forms an RNP that starts scanning the chopped pieces of foreign DNA. The Cas9 specifically looks for a short 3-base NGG sequence. This is called the PAM, short for protospacer adjacent motif. The PAM interacting domain in Cas9 performs the PAM scanning function. And when it finds NGG, the immediate upstream stretch of DNA is destined to become a protospacer. Hopefully, now you notice the connection between protospacer and its adjacent motif. In this scanning mode, the Cas9 RNP is not active. The only function Cas9 here is doing is to find the PAM region. In some capacity, CSN2, which is a ring-shaped protein, may help in sliding Cas9 on the DNA, although its exact function is unclear. But when the NGG is bound, a set of proteins that I didn't draw in the picture above take over from here. This complex has 2 Cas2 proteins, forming a dimer. Cas2 dimer bridges 2 Cas1 dimers, 1 dimer on each side. Both Cas1 and Cas2 are endonucleases. The PAM recognition essentially triggers the trimming of the protospacer from this long stretch of DNA where Cas9 is bound. The Cas1-Cas2 complex cuts out 20 to 50 bases long DNA, which will be integrated into the CRISPR locus in the next step. One important note, the trimming is done exactly before the N in the NGG. The final 20 to 50 nucleotide spacer that is cut out does not include the NGG sequence. With this, the bacteria has acquired a new spacer, and let's call this S0. The next step is to integrate the spacer into the CRISPR locus. The preferred spot to integrate the spacer is after the leader, where you have a short leader adjacent sequence, right before the start of the first repeat. This LAS attracts the Cas1-2 complex. The Cas1-Cas2 complex causes an unusual bending of the first repeat, which in turn exposes the grooves of the repeat DNA, allowing the endonuclease domains to cleave the first repeat on the opposing strands, while also unwinding the repeat through an unknown mechanism. Essentially, the repeat 1 splits into 2 strands, and while this is happening, the spacer is ligated in between the split repeat 1. The missing strands of the repeat likely undergo a gap repair step. So we have just duplicated the repeat on both sides and integrated a new spacer S0. The mechanism of how all this happens isn't that well characterized. Plus our mystery protein CSN2 is somehow also involved, but we don't quite know the full extent of it. Now that we have got a new spacer, the bacteria needs to use this memory. The first step in the retrieval is to make RNA from the CRISPR operon. The entire CRISPR locus is converted into a giant, non-coding precursor CRISPR-RNA. On the other side, the tracer operon is also transcribed to get a complex trans-activating RNA, which we saw in the spacer acquisition as well. The Cas9 protein is also part of this process, and I think now is a good time to expand on this structure. The Cas9 protein is bilobed. One lobe does the recognition job, the other one has a nuclease function. There are 2 nuclease domains in that nuclease lobe. One is a RuVC-like nuclease. You may have heard of RuVCs in holiday junction resolutions. The other nuclease is a homing endonuclease. And you've already seen the PAM interacting domain. Take note that in its default state, the Cas9 protein is inactive. The HNH nuclease domain inhibits the RuVC domain. We saw this inactivity in spacer acquisition as well. Now the CRISPR-RNA biogenesis can begin. The tracer-RNA has partial complementarity to the repeat region of the precursor CRISPR-RNA. The tracer-RNA therefore decorates it wherever it can find a repeat match. This repeat match allows Cas9 binding to stabilize onto the CRISPR-RNA. The Cas9 does not wait for tracer-RNA and CRISPR-RNA to pair. The Cas9 can come preloaded with the tracer-RNA. The pairing merely stabilizes this complex arrangement. As the Cas9 is stabilized onto this complex RNA, the RNase 3 cuts the CRISPR-RNA near the end of the repeats. This releases a spacer repeat Cas9 complex. meaning that each Cas9 RNP now carries one specific spacer. This spacer can be S0 if it comes from this chopped part, it could contain S1 if it's from this portion, and S2 from the next one, and so on. This digestion by RNase 3 is the start of a maturation step. The maturation proceeds with an unknown exonuclease that eats away the protruding parts of a spacer. The 20 to 50 nucleotide spacer is trimmed down to right around 20 nucleotides. This RNP is a catalytically competent Cas9. To make it explicit, it is a 3-component system of Cas9 protein and 2 non-coding RNAs, the tracer and CRISPR-RNA. The CRISPR-RNA has exactly one spacer of 20 nucleotides and exactly one repeat paired with tracrRNA. After retrieving the spacer in the form of CRISPR-RNA, it is now time to use it as a weapon. So imagine that the same enemy comes around to harass the bacteria. This time, the bacteria recognizes this attack based on the spacer. Since it can retrieve the spacer DNA from its library, the Cas9 scans the foreign DNA to find a match at the S0 segment in the pathogenic DNA, guided with the help of CRISPR-RNA. But the process of guiding Cas9 to the correct location starts with the PAM interacting domain, which is looking for an NGG sequence. Recall that NGG was used in memory creation as well. This step is called PAM verification. There could be many NGGs in the foreign DNA, but only one specific NGG will be next to the S0 spacer. When an NGG is sampled, the RNA loop, which is the spacer portion of the CRISPR-RNA, tries to base pair with the DNA immediately next to PAM. This will happen with all NGGs, but only one specific location will find a match with the spacer S0. When the other NGGs are sampled, the R loop will fail to prime and Cas9 will not proceed with the next step. Let's talk about sampling and R loop priming a bit more. When the CRISPR-RNA tries to pair with the DNA, the first 10 to 12 nucleotides, called the seed region, should be a perfect or a near-perfect match. When the seed region pairs, the R loop expands, meaning that the unwinding of DNA extends, and the remaining 8 to 10 nucleotides of CRISPR-RNA base pair. Unlike seed regions, they do not need to be a perfect match. Just to recap, the seed region primes first, and if it does so successfully, the priming extends and the loop expands by base pairing. This, in turn, makes the HNH domain catalytically active, which allosterically regulates activation of RUV-C domain. This initiates the final step, DNA cleavage. The target DNA strand, which is complementary to the guiding CRISPR-RNA, is cut by the HNH domain, and the other strand is cut by the RUV-C nuclease domain. More specifically, the cut is made 3 nucleotides upstream of the NGG, and the cut is made only in the DNA strands, not in the 20 nucleotide spacer corresponding part of the CRISPR-RNA. Since it is 3 nucleotides from PAM on each DNA strand, this is a blunt, double-stranded break. This cleavage of foreign DNA marks the completion of bacterial defense. It is the bacteria's way of making a vaccine against foreign DNA elements. This interference step, the RNA-guided DNA cleavage, is repurposed as a tool for targeted genome editing. A video on that will follow soon, and I will see you there.